Perfect. So thank you all for being here. Like I mentioned last week, the main purpose is mostly to introduce myself to the group in terms of what I do with Benthic forams in terms of research, with the hopes that this could lead to potential collaborations in the future between nematodes. I mean, working with nematodes and Benthic forams. So what we have here on this SEM plates, here we have just a healthy individual of the genus Ammonia, and then here we have another from green colored lina, and then here we have the deformities associated with them. In the old days, it was thought that deformities is related to pollution, but it's a hard question to answer. Deformities can be triggered by anything, changing oxygen, texture, salinity. So nowadays, there's no direct correlation between deformities and pollution. But with that said, I do culture ammonias. And that's what we have here on the left side. Laser point, here we go. That's what we have on the left side. This is a work in progress. And the idea is that when you culture ammonias, or any calcareous foram, in the presence of copper can substitute for calcium during biomineralization. So the hypothesis is that translocation from copper to calcium might create a distortion in the crystal lattice of the calcite, which might potentially trigger a deformity that you might see in field samples or in culture samples. So this is just a work in progress related to culturing living ammonias. Why this thing is not moving? Here we go. So again, overall is just to share with the meiobenthologists attending today what I do with Benthic forams, and hopefully by the end we can all chant that forams rule as the best creator ever for research. And so as we all know, estuaries, they receive a lot of waste. I work particularly in estuarine environments, this transition between land and sea. I'm a geologist by training. I have a PhD in marine science, so I do ecology, paleoecology. So depending on my question, I can look at individuals, a single species, or typically I work with communities. So that's nothing new. But everything is related to mostly heavy metal pollution, and more specifically in terms of bioavailability of heavy metals to the Benthic foram community. So for the sake of those of you who doesn't know what forams are, so these are protists. However, they do have a shell. So these are shell protists. They have different types of feeding strategies, the frugivores, carnivores. Some host symbionts, and during the lack of proper food, they will feed off their symbionts. Other critters or forams, they're going to go kleptoplasticity as well, like the genus Ophidium. They harbor kleptoplasts. They do engage in rapid sexual reproduction as well as in asexual reproduction. So if you're culturing with one individual, you can get somewhere between 50 to 150 new baby forams. So when I'm culturing, that's what I do. I try to force asexual reproduction so I can have enough individuals for my culture work. The only issue that I have right now is trying to find living forams. Here in Florida, in environments that I work in estuaries, again, it's been challenging in finding living ammonias for culture work. Granted, I just need one or two. And even though when I find one or two living ones, it's really hard to get them to reproduce. So it has been challenging. So typically I rely on outside colleagues from Europe to ship sediments that are riddled with ammonias for culture work. No, it doesn't work. So forams, you have two main varieties in terms of their shell. So you have the calcareous and then the agglutinated. So the calcareous, they biomineralize calcium carbonate, and you have two main varieties. You have the hyaline, it's basically the transparent or translucent. So this is an ammonia genus. But then you have the other ones, which are the meiolytes. So think of a milky white shell. But also then we have the agglutinated. So these ones, they don't biomineralize calcium carbonate. They just rely on the sediment particles in the sediments, and then they do precipitate calcium carbonate as a cement to create their shell. And then we have the ones that have the organic wall, so like monofilaments. And these ones is the ones that I have the least experience with, because it's the way I process samples. Since I work with total assemblage, basically it's living and dead combined. The way I process the samples, these monofilaments, they get destroyed. And also when I'm looking on the microscope, I'm looking at oven dried sediments. So these are long gone by the time I sprinkle sediments under the microscope. I keep forgetting. As nematodes, you have some species or genera that are susceptible or resistant to environmental changes. So this is a modification of Jorgensen's Trucks model. Here we have depth, and then we have eutrophic, mesotrophic, and oligotrophic conditions. And this is just to illustrate how the assemblage of a benthic foram will change based on the amount of organic matter and the amount of dissolved oxygen present within the sediment water interface, or even within the pore water. And they are ubiquitous. They're found everywhere. This is a nice paper. I posted it on Slack from 2012. They were comparing benthic forams and nematodes when it came to pollution. And again, from an ecological point of view, it's very helpful. You have increase or decrease in density, diversity based on proximity to the source. In the case on, I'm preaching to the choir with the nematodes, is based on the maturity index. But also you have, as you know, species that are resistant or tolerant or sensitive or oversensitive to contaminants. It doesn't have to be heavy metals. It could be, again, dissolved oxygen, a change in texture. And then they are ubiquitous. They're everywhere in the oceans. We have the freshwater variety of benthic forams, and there was a publication that came out maybe like 8-9 years ago by Pawlowski and his group, in which they found DNA of Foraminifera in soils. So pretty cool that we have a potentially a terrestrial variety of forams, benthic forams. So when it comes to forams, typically, as a rule of thumb, we expect to find most living individuals within the first 2 centimeters. So again, this is a rule of thumb. When we are working with sediments, that's more or less our sampling resolution. I mean, granted, you have some species that are going to be deeper than that, but it's just a handful of them. And depending on your question, some researchers, they do a 1 centimeter sampling resolution, others do 0.5 centimeter sampling resolution. But it's what accepted within the foram community that within the top 2 centimeters, you should have the bulk of the living assemblage. So with the benthics, you have, based on morphology, epifaunal, infaunal, and of course, epiphytic. And one advantage of the benthic forams is because they have a shell, so they get preserved in the rock record or sedimentary record. So if you're looking at sediments that are 10 million years old, and if you study the actual morphology or the shape of the shell, you can tell, oh, these were epiphytic, these were infaunal, these were epifaunal, whatever it is. So in other words, the morphology of the shell is related to the type of habitat or niche that you're going to have those forams, benthic forams. And the question is, are they meiofauna? I'll say yes, of course, but according to Fenchel and Higgins, for something to be considered meiofauna has to be within that size range, between 50 and 1,000 micrometers. Standard benthic foram studies is you see the sediments through a 63 micron. So anything that's left over, you're going to find forams, so it falls within that size range. However, some of the researchers, they prefer the 125 micron size as well, so it does fall within that range. So as far as I'm concerned, sure, of course, benthic forams, they are meiofauna. So here we have 2 living ammonias ganging and feeding on a live copepod. So again, the detritivores, the carnivores, bacterivores as well. Same authors, they found the same genus feeding on a nematode. as you can see on this image from their publication. So they're quite voracious feeders, believe it or not. When I culture them, I feed them dunalielas, which is a green algae, and I have to feed them every 3 days. So they're quite voracious feeders, or eaters, sorry. They'll eat anything basically that falls within their reticulopodia. So this year, this is one great advantage that forams, benthic forams, and planktonic as well have. So these threads that you see here on this image, this is granular reticulopodia. It's basically modified sugars. And they use this for motility, feeding, reproduction, and calcification. So it's a multi-purpose, and I'm saying this loosely, appendages that benthic forams use. So what is the ecological theory behind benthic forams? Well, I mean, similar to nematodes, they're sensitive, so they're going to be fast responders. Precise, they're predictable. In other words, if you know the assemblage of a pristine environment, and then you have some type of pollution coming in, or if there's an environmental natural change, then you should expect to see, well, if we have this assemblage here, then we should expect the assemblage to change to this other type. So it's predictable in terms of the, to some extent, it's predictable in terms of their ecological response. Because they have a shell, they're great in assessing and identifying preconditions or pre-impact conditions or pre-management conditions. So this is one topic that I am currently working on with collaborators from France, Switzerland, and in Florida, is to, we're gonna take sediment cores and we're gonna, again, implement and assess if we can identify pre-management, pre-impact conditions back in time. And of course, when it comes to the physiochemical characteristics of the seawater, they're sentinel species, as there are a lot of research done in culturing forams at different PCO2 levels, just to have an idea of what will happen to this planktonic foram specifically with an increase in ocean acidification. And of course, they have been used and still being used for paleoclimatic reconstruction. So they're quite diverse in terms of how you can use them, the forams. So I like this quote from Sengupta. In the old days, is basically, we use the environment to understand the assemblage, but since the 50s is, let's use the assemblage to understand the environment. And that's when environmental micropaleontology started, with that paradigm shift since the 50s. And that's what has been done lately with fairly good success in terms of assessing the levels of environmental impact, environmental health of coastal marine as well as deep water systems, just by a paradigm shift in terms of how will you use and assess your assemblage of the benthic foram. So some of you might be scratching your heads and say, you know, WTF, I get it. And I agree with you, what is their functionality? So I'll try to answer that question with this. Here we have two genera. So this is the genus Ophidium, and here we have Ammonia. This genus is, they proliferate in oxygen-rich environments. Ammonia is the opposite. So when you look at the relative abundance of the ratio, you have what is known as the Ammonia Ophidium index, and this is was developed by Sengupta. And this is a study that got submitted from Brazil. And we have this long core, about a 1,000-year-old record, sedimentary record. And we were looking at foraminifera assemblages, heavy metals, changes in texture and whatnot. And I'm just going to highlight the top 40 centimeters. So this is the Ammonia Ophidium index. So for the past 8,000 years, these two were slightly present up until 1975, where we start seeing this increase in the index, which tells us, okay, there's a change in oxygenation since 1975. The question is, okay, what is causing this? Is this just a change in the watershed in terms of organic matter influx? Does it have to do with dredging activities. And in this bay, Sepetima Bay is a combination of a little bit of everything. This FSI is a FORAM stress index, which uses different species to assess, again, environmental health or degradation. So we're seeing that post-1975, we see an environmental degradation of this estuary. We see a decrease in oxygenation. And then we have the sediment pollution index based on heavy metals. So we do have a correlation between the amount of contaminants coming in and the ecological response of these benthic forams. So since we saw this, that there was a decrease in oxygenation, well, let's look at then at some of the heavy metals. I do a lot of do geochemistry, sediment geochemistry. So we were looking at molybdenum uranium ratios as seen here, and then we just did this plot, and it's for me it's uncanny. I just want to highlight here. So this is the bottom sample, 174, then 166, and then 150. So as we're moving up core between 174 and 150, we're moving towards more anoxic settings. Granted, this was a sub-aero environment during that time, so we're assuming that we have some lacustrine conditions. Then, of course, no benthic forams. And then from over here, we go from 148, then moving our way downwards, in which then oxygenation is increasing. So we interpret this as this environment going from terrestrial into fully marine setting. And then between 40 centimeters, which is where we started seeing this impact on change on oxygenation and pollution. And notice how uncanny, how these samples, they just migrate with depth, shallowness, indicating that you do have, again, another change in oxygenation. So here we have a mixture. This particular bay, it was terrestrial, then it became fully marine, and then it became restricted. And that's when estuarine conditions began to develop. So we have a natural environmental change toward estuarine, but then we have an increase in organic matter, and then of course we have the impacts or effects of dredging, which is again reflected on these two biotic indices when we implement benthic forams. So I'm also collaborating with a colleague from Nigeria. So this is Lagos Lagoon. So here we have the surface distribution of these two, which are the agglutinated ones. So they take sediments to create the shell. This is ammonia that biomineralized calcium carbonate. So what is really nice about this is when we did the clustering of the samples based on a cutoff on organic matter. So we have these sediments, these stations clustering when you have less organic matter, and that's where you have ammonia dominating the assemblage. So that's the genus that precipitates calcium carbonate. We have these other samples that are more enriched in organic matter, and the benthic forams dominating are the ones that take sediments, surrounding sediments, to create the shell. So we just try to correlate their distribution with the several heavy metals. And one point of contention is that a lot of researchers, they use total heavy metal concentrations. That helps if you're working with organisms that have a digestive system. Forams, they don't. So it raises the question, so if you have a high concentration of heavy metals in the carbonate fraction, would that be bioavailable to the foram if it's within the crystalline structure? Or the same with iron oxides. So we sequentially extracted the heavy metals, and I'm just showing this one here. This is the organic fraction. So this is chromium, cobalt, copper, and nickel within the organic fraction of the sediments, and this is distribution. So we have some hotspots, which kind of relates to some of the hotspots of the distribution of these forams. But when we look at the statistics, we have ammonia is the only one that's negatively correlated with the cobalt within the F1 fraction, meaning the cobalt that it is adsorbed to the surface of the sediments. Which makes sense because one hypothesis is that heavy metals adsorb to clay sediments and heavy metals complex with organic matter are hypothesized that those should be the most bioavailable to the forams. But when we look at Trocamina, it's basically correlating positively with every single fraction. And that's where the question comes in. So F3 is heavy metals associated with iron oxides within that crystalline structure of iron oxides. They don't ingest sediments. They don't ingest iron oxides. So that's the question, what is bioavailable? So this here is a point of debate. What is bioavailable? So just because this correlates positively with every single heavy metal fraction doesn't necessarily mean that it is bioavailable. So we're looking at here at false positives. So that's one thing that my students and I are working in my lab, is to provide more lines of evidence to show that discrepancy between comparing total heavy metal concentration, what's bioavailable, with foraminiferal distributions. So this is an X-ray from Puerto Rico. So we took sediment cores, and we were lucky to get a time frame with lead cesium. And I just want to highlight down here. Here we have this spike in an increase in sedimentation, an increase in mud content. Here we have, of course, a decrease in organic matter. So that tells us that there was a change in the watershed, a change in the provenance of the sediments. And we use rhenium as a proxy for paleonoxic environments. So when you have a rhenium profile and we have a sharp increase in rhenium, that's indicative that is precipitating under anoxic conditions. So we were able to interpret this change in rhenium concentration as anoxic conditions starting around 1930s, 1950s, or prior to that, sorry. And then above that, then you have your standard normal well oxygenated conditions. So how does then this relates to metals? I'm just gonna show copper. And copper does follow that same pattern of clay sedimentation. Copper, zinc, cadmium, nickel had the same profile. So that just tells us right there and then that these heavy metals are readily adsorbed to the surface of these clay sediments, which should be more bioavailable to the benthic forams. So what does the forams look like in the same cores? So when we have the anoxic conditions, like I mentioned before, ammonia thrives under lower oxygen conditions. So we have a slight increase in their relative abundance. Once we hit or find normal oxygen conditions, notice how they decline in relative abundance. So that's, again, that's why this is very useful as an indicator of anoxic conditions or changes in dissolved oxygen. The meiolis and like quinquecolinas, for example, is the opposite. They thrive more when you have better oxygen conditions. So down here for our stats, this means total copper concentration, and this is bioavailable concentration. So notice that nothing for this particular forams, nothing correlated with what's supposed to be bioavailable. But this is bioavailable copper associated with calcite. And this is great because it shows that you're getting false positive and false negatives. People will say, oh, copper, when there's more copper, there's more ammonia. Well, not really, because what it was supposed to be bioavailable is not correlated. So you are overestimating when you work with total concentrations of metals with benthic forams. With nematodes, I'm assuming that's not the case because they have a digestive system. So if they are ingesting sediments, they might be able to process, and please correct me if I'm wrong, they might be able to process iron oxides or something like that within their digestive system, which in the case of the forams, that is not the case. So that's what I'm currently aiming in my research lab, is to deconvolute this issue with total heavy metal concentration and what is bioavailable to benthic forams, since a lot of studies are coming out using forams as bioindicators of heavy metal pollution, but for the most part, they're using total concentrations, not what is bioavailable. So I've also worked with coral reefs. This is a study that we did in Belize, not with heavy metals. We were looking at the effects of sewage on coral reefs. So between this island in Belize, this is a reef, it's about a mile and a half in between. So what we have is a FORAM index. So the FORAM index, it's a Benthic FORAM index designed specifically for coral reefs. It's an indicator of water quality. So you have oligotrophic, mesotrophic, eutrophic, and hypertrophic conditions. Number of species when it comes to FORAMs, medium, high, and of course mesotrophic is kind of a Goldilocks conditions. And when you have this type of conditions or environments, then the FORAM index does relate to, if it's oligotrophic, you need to have values greater than 4. If it's hypertrophic, then you're going to have very low values. So when we implemented this in Belize, we see that most of the sample stations are above 4. So very good water quality. So where is the sewage coming from? This island in Belize, they don't have any treatment facilities. When you flush, it goes down to the coastline. So, and I'm gonna say this loosely, what we thought is that it's just getting diluted because it's almost 2 miles away between the shoreline and the reefs. Yes, you're gonna argue, but you have very low FORAM index values, which is indicative of hypertrophic. Well, in these particular stations, the low values of the FORAM index are linked to a change in substrate. So in these particular sites, it was barren. It was just a hard substrate. It was no sediments. So the amount of reef-dwelling FORAMs were very low. So it wasn't not an effect of sewage. It was just a natural effect of a change in substrate. This publication, then, we went back and reinvigorated the FORAM index, comparing Caribbean settings with Pacific Ocean. In other words, the FORAM index was developed for Caribbean and Gulf of Mexico subtropical conditions. So when you're implementing it elsewhere, like with any index, you need to be very careful. It doesn't translate on a 1-to-1 ratio. You need to consider other reef-dwelling FORAMs. And that's what we try to illustrate in this publication, is that you have the carcharenids. The carcharenids, which are these star-shaped looking benthic FORAMs, they are there, so they need to be reassessed when it comes to implementing the FORAM index. The FORAM index formula is based on the type of FORAMs that harbor symbionts, that are heterotrophs, and that are stress tolerant. So that's what you have here down in the formula. With the carcharenids, there's evidence in the based on previous work that carcharenids, you can find them in naturally stressed conditions. So that raises the question, and even though it's a reef-dwelling critter, it raises the question of, how transferable is the FORAM index outside Caribbean or subtropical settings in the Gulf of Mexico Atlantic. So that was the purpose of this article, and also to provide guidelines on how to proper process sediment samples, so on and so forth. Now we're going to be moving a little bit towards deep water. So I guess you might be aware of the AMBI index. So since 2016, the AMBI index has been implemented using benthic FORAMs. So it's called the FORAM AMBI index. And it's basically you're looking at the relative abundance of benthic FORAMs, or if you're doing the regular AMBI, which is on micro inverts. With benthic FORAMs, you're just plotting them against the amount of organic matter in your samples. So this is semi-quantitative, semi-qualitative approach, but it has been used a lot, the AMBI, and now we're testing the FORAM, excuse me, the FORAM AMBI index in the Gulf of Mexico. The formula is the same as standard AMBI index based on the actual distribution of your FORAMs. Then you make an assignment to which group it falls into. And then this is a heat map generated by O'Malley. He's a grad student of Patrick Schrein from University of South Florida in Tampa, St. Petersburg, sorry. So this heat map is just showing you, of course, the ecological quality status based on the FORAM ambi as a baseline. So that was the main objective, is just to generate those baseline conditions based on the relationship between benthic forams, deep water benthic forams, and organic matter distribution. So I hope you're understanding that I mainly concentrate in shallow water, coastal, estuarine conditions, but I also work on coral reefs, and I've also collaborated in deep water settings, but my main forte is it's shallow water estuaries when it comes to benthic forams. So what are the criteria for the use of benthic forams in environmental studies? Like mentioned previously, and similar to nematode, they're sensitive, so they're fast responders to stressors. And the fact that they can reproduce very fast asexually and produce so much offsprings, understanding and studying their ecological response is very, very helpful. So they're precise in terms of the predictable, in terms of their ecological response to environmental change. And the question's always there, you know, if there's an environmental change, how can we deconvolute if it's a natural environmental change or an anthropogenic environmental change? So that's what you need to rely or use of the proxies like heavy metals, for example, to assess if what we're seeing is anthropogenic in nature, simply from looking at heavy metals. But also, if you look at aluminum is a good proxy for terrestrial input of sediments. So it's not only using heavy metals as a pollution indicator, you can use other metals, aluminum, manganese, titanium, for example, as terrestrial proxies of sediment input. So you might also use that to help you deconvolute whether what you're having is natural or not. What applications? Spatial temporal, like I showed you before, those spatial maps, and then within a core. They're representative, so they're good model to follow with respect to other responses, again, whether it's natural or not. They're integrators. They will incorporate the environmental physiochemical parameters, not only from the point of view of paleoclimate reconstructions, but also when it comes to pollution studies, you can also assess the levels of copper, nickel, and whatever heavy metals you're looking at within the test or shell of the benthic forams. And they're tattletale, so they can foreshadow changes in the environment. So if you have your standard monitoring sites, and that's one thing that we're trying to implement in the state of Florida with the Florida Department of Environmental Protection, is to see if they will integrate the use of benthic forams in their monitoring sites. So then you can have a good idea, because they're sentinel, they're fast responders, then they should be able, if the forams are showing a change, a negative change, then resource managers should be able to act accordingly. So for example, the FORAM index is a water quality indicator. The main benthic foram used in the FORAM index is the ones that harbor symbionts, like Amphistogina, the genus Amphistogina. Amphistogina, they bleach like coral reefs. And actually there's an Amphistogina bleaching index. And it has been found that Amphistoginas or reef or any other symbiont-bearing foram in coral reef, they bleach a couple of weeks before you see a bleaching event in coral reefs. So this FORAM index was implemented in the Great Barrier Reef in Australia from 2012, if I recall, up until 2016, 17. And then they discontinued because of lack of funding. But you have that rapid response. So that's why they're good tattletale. They can foreshadow a major environmental change just by looking at the microenvironment of the benthic forams. And other than that, this is, recently we found this microturbularias, cocoons attached to living and as well as dead benthic forams. This is from Apalachicola Bay here in Florida, close to where I work. So, so we're working on this. There's a colleague that he does molecular work on microturbularias. So, because I didn't know what they were, I thought they were parasites. And then searching, I discovered, okay, these are flatworms or micro flatworms. So I'm just right now hunting for not hatch cocoons, but unhatched cocoons to send to him so he can do molecular work, so we can get a taxonomy. And then from there, start thinking, okay, why then they're using benthic forams? Although I have found the cocoons attached as well on quartz sand grains, but most are found attached to forams. So in this particular bay, forams seems to be the preferred substrate for the microtubularia. So that's a question that needs to be answered. Specifically given that in this bay, and this is the genus Ophidium, and these are agglutinated forms. Because this bay is an estuary, it's a river influence estuary, so you have big gradients in salinity. So the abundance of benthic forams is low. For an estuary, is very low. So even with that, forams are the preferred substrate for the cocoons. So that's a work that is ongoing. So if any of you have any ideas and you're interested in collaborating, by all means, just let me know. Shoot me an email or through Slack, and we'll continue with this. But this is a work in progress. And aside from research, I like baking. So I always told my wife and my advisor, if I don't get tenured, I'll open a bakery. And so I guess I can get by making cakes and pastries if forams don't pay out, you know. And that's it. So I hope, again, you understand that main purpose was just to introduce myself, what I do, maybe trigger ideas for potential future collaborations. And I'm open, very flexible. I know Jerome for the last 3 years since I started working at my current position at FAMU. So we're collaborating as well, him and I. So again, thank you for your time. And let's see questions.